I hope everybody is able to see the screen. I
Yes,
will
absolutely.
stop my video for just bandwidth's sake. Um, a very good evening to one and all. My name is Pavana Rotti, and I will talk about how lack of CFTR alters the pancreatic ductal secretome and proteome and its effects on endocrine and exocrine signaling in the pancreas in CF. This is a part of my PhD work at University of Iowa. So the pancreas is both an endocrine and exocrine gland. The exocrine gland consists of the ducts and acini. The ducts form a lumen into which the acini secrete digestive enzymes. The endocrine pancreas consists of islets formed by these 5 endocrine hormone secreting cells. Of these, the beta cells secrete insulin, and when blood glucose levels rise beyond the normal range, the beta cells secrete insulin. Insulin enables, insulin enables glucose uptake by cells to reduce blood glucose levels. This ability to regulate systemic glucose levels is called glucose tolerance. And CFTR as an anion transporter is expressed predominantly in the ducts. Now, CFTR, an anion channel, is expressed in the pancreatic ducts and present on the apical membrane of the ductal epithelium. CFTR transports bicarbonate either directly by permeation through the channel or indirectly by regulating chloride and bicarbonate exchange. So functioning CFTR ensures alkalinization of the ductal lumen. And this is essential to maintain the acinar secreted digestive enzymes in an inactive form. Now, with the loss of CFTR function in cystic fibrosis, the ductal lumen is acidified, and this activates the digestive enzymes while they're still in the pancreas, and they begin to digest the tissue, causing tissue necrosis. So cystic fibrosis related remodeling occurs in both endocrine and exocrine pancreas. Histological analysis of pancreatic sections from CF patients have revealed the extent of pancreatic remodeling. Now, acinar atrophy accompanied by adipose tissue infiltration is very prominent in CF pancreas. The islets are found surrounded by adipose tissue. The mucus obstructed ducts are surrounded by fibrotic tissue, and the islets are found coalesced amidst fibrotic tissue very close to the ducts. As a result of CF-induced remodeling in the pancreas, about 85% of CF patients are pancreatic insufficient, and 50% of the adult CF patients about the age of 30 years are diagnosed with cystic fibrosis related diabetes, which will further be referred to as CFRD. Impairment in endocrine pancreas is postulated to be due to the bystander effect, which is primarily due to ongoing pancreatic and exocrine remodeling. However, in CFRD, pathophysiology is unique and not very well studied. It is distinct from type 1 diabetes in that reduction in insulin secretion occurs without significant loss in beta cell mass. CFRD pathophysiology doesn't resemble type 2 diabetes, as insulin resistance is not observed in CF patients. Hence, understanding molecular determinants of CFRD pathophysiology is important and is enabled by CF animal models. So animal models used to study the effect of CF in various organs include the traditionally used rodent models like the mouse and the rat. But pigs and ferrets have gained more prominence in recent years for their resemblance to the CF-induced pathology observed in humans in various organ systems. So CF rodent models show little to no damage in the pancreas. The CFTR knockout or CF pigs are born with extensive inflammation and severely damaged pancreas. The CF knockout pigs are born with severely damaged pancreas, so it's difficult to study the evolution of the disease in pigs. However, CF ferrets, on the other hand, are born with little to no damage in the pancreas, but rapidly progress through stages of pancreatic remodeling, resembling that seen in human CF patients. So what changes in the CF? What are the observable changes in the CF pancreas in ferrets? Histological analysis of hematoxylin and eosin stained pancreatic sections from wild type and CFTR knockout ferrets revealed features that were consistent with CF-induced remodeling in human pancreas. Newborn CF ferrets had normal pancreas with abundant acini and undilated ducts. They, however, had abnormal glucose tolerance at birth, even with intact pancreas. This stage is referred to as the newborn stage. At around 1 month of age, the pancreas of CF knockout ferrets developed multifocal inflammation with slightly dilated acini. However, their glucose tolerance remained abnormal, and this stage is referred to as phase 1. At around 2 months of age, pancreas of CFTR knockout ferrets had mucus obstructed and dilated ducts surrounded by fibrotic tissue. The islets were found coalesced near these ducts amidst fibrotic tissue. Ferrets underwent a phase. These ferrets underwent a phase of glycemic crisis at this age. And further, at 3 months of age, pancreatic pathophysiology of CFTR knockout ferrets continued to worsen with near complete loss of acinar tissue and adipose tissue infiltration. The ducts were dilated and mucus obstructed with coalesced islets close to them in fibrotic tissue. Remarkably, the CFTR knockout ferrets at this stage recovered from their glycemic crisis. Now, in addition to age-related changes in glycemic tolerance and local inflammation and adipogenesis were analyzed. Inflammation peaked during the period of glycemic crisis and decreased by 3 months, but adipogenesis and fibrosis peaked at 3 months and remained high. At 5 months of age, CFTR knockout ferrets developed cystic fibrosis-related diabetes as a result of beta cell failure. A similar transient recovery from abnormal glucose tolerance is observed in CF patients between 5 to 10 years of age. Hence, we focused on characterizing alterations in pancreatic duct and endocrine cells during the glycemic crisis period preceding to the recovery and up until the recovery phase, which is between the phases phase 2 and phase 3. Now, in order to understand the changes in the endocrine pancreas, we stained the wild type and CF ferret pancreatic tissue sections with endocrine hormones like insulin, glucagon, somatostatin, and quantified their abundance using morphometry. There was a significant increase in the frequency of endocrine hormone expressing cells between phase 2 and phase 3. Again, this is where this is the phase during which recovery in glucose tolerance happens. Additionally, the diameter of the islets increased significantly in CFTR knockout ferrets compared to the age-matched wild types when normalized to the entire pancreas. So the ducts in CFTR knockout pancreas were found associated with endocrine cells in the period of severe inflammatory insult, which is at around 2 months of age or during phase 2. And the ductal lumen during this stage also contained a significantly higher number of Ki67 expressing cells or proliferating duct epithelial cells. Now, given the increase in endocrine cell frequency and the presence of endocrine cells in CF duct epithelium during the phase of severe inflammatory insult, we hypothesized that there were altered exocrine signals from the duct that initiated this change in the pancreas. So in the context of CF-induced injury to the pancreas, in the presence of proliferating duct cells, activated fibroblasts and inflammation, are there paracrine signals from the duct that affect islet function and islet abundance? And do the paracrine signals from this altered cellular composition of the pancreas affect disease progression? How does CF-induced remodeling affect ductal phenotype? Now, in order to evaluate this, we developed an in vitro ductal cell primary cell culture system. We harvested pancreas from wild type and CF ferrets in the neonate stage, digested them, and produced long-term proliferating ductal cell cultures in conditionally reprogramming media. These cells were called proliferating ductal cells. These were then later polarized at air-liquid interface to establish the apical and basolateral boundaries, and they were differentiated or polarized in the presence of media which does not contain any ROCK inhibitor. And at this stage, these cells were called the pancreatic duct epithelium. So when at the pancreatic duct epithelial stage, we characterized both wild type and CF pancreatic duct epithelia to see if they expressed the key ductal markers like SOX9 and KRD7. And as you can see on the left, they did. Both CF and wild type cells expressed both SOX9 and KRD7. We also evaluated how the function of CFTR in the pancreatic duct epithelium. These polarized cultures were induced with IBMX and forskolin, which increases intracellular cyclic AMP and stimulates CFTR function. And we saw that only the wild type cells presented with CFTR function, while the CF cells had no CFTR function. In order to establish its physiological relevance, we also checked for apical pH of both wild type and CF pancreatic duct epithelium, and saw that the wild type cells were indeed more alkaline compared to the CF duct epithelium. Now, in order to evaluate how CF ducts affect islet function, we decided to quantify the differential proteome in the CF cells and the differential secretome, or the proteins secreted by CF cells that were different compared to that of wild type. And for this, we used both wild type and CFTR knockout polarized duct epithelium and stimulated both of them with carbachol or IBMX on the basolateral side and collected the apical and basolateral secretions. In addition to this, we also harvested the cellular proteome. So with these samples, we used stable dimethyl isotope labeling or quantitative mass spec analysis to quantify the relative abundance of CF samples with respect to the wild type samples as a reference, and calculated the fold changes in CF with respect to wild type. The apical secretions contained in CF contained 28 differentially secreted proteins, whereas the basolateral samples had 7 differentially secreted proteins. Now of these, some prominent ones were IGF binding protein 7, S100A11, SPARC, and DPP4, which are known to have an impact on islet maintenance and development. In fact, about 30% of the differentially secreted proteins, or the DSPs, were previously associated with islet maintenance. Now, using ingenuity pathway analysis, we were able to find the upstream signaling regulators of differentially secreted proteins in CF, and top hits turned out to be TGF-beta signaling, Wnt, BMP, and Notch, all of which have a role in pancreatic and islet developmental programs. Shown in the heat map are, the p-values for the signaling ligands, and this one here is the activation z-score. A value of activation z-score lesser than -2 indicates suppression of the pathway, whereas a value greater than 2 usually indicates activation. We also map the differential interactions between the various cell types in the pancreas. The differentially secreted proteins were mapped to their cognate receptors, and the cells those receptors are enriched in. This gave us a list of putative paracrine interactions, and we found that the duct signaled to both the endocrine and exocrine pancreas. So the duct to acinar signaling was about 23.8% of the total interactions. Duct to duct was about 28.5%. Duct to endocrine and duct to stromal was 23.8% each. This indicated that the differential secretome from CF ducts affected both endocrine and exocrine pancreas. We also verified if the differentially secreted proteins found in vitro also varied in vivo. What I show here is one candidate differentially secreted protein, IGF binding protein 7. For this, we used single molecule in situ hybridization and saw that the ducts in the CF ferret pancreas expressed lower IGF binding protein 7 as we observed in our secretion assay in vitro. But the surrounding fibroblasts, but as you can see here, the surrounding fibroblasts had increased expression of IGF binding protein 7, and they also infiltrated into the islet regions here, which indicated to us that in CF pancreas, islets were exposed to an excess of IGF binding protein 7. So to recapitulate this, we exposed wild type islets to IGF binding protein 7 and saw that the profile of insulin secretion from the exposed islets was similar to that observed in CF islets, in that in the presence of low glucose, insulin secretion was higher, which means the basal level insulin secretion was higher. And in the presence of high glucose, when exposed to IGF binding protein 7, insulin secretion was decreased, opposite to what is seen in normal islets. And this indicated that ductal secretions in part impact islet function. So now moving to the whole cell proteomics data, we found 303 significantly altered proteins in the CF duct cells, out of which 31 were downregulated and 24 were upregulated. We identified the proteins that directly interacted with the CFTR protein to analyze the proteome differences directly associated with the absence of the CFTR protein on the membrane. So about 3.5% were extracellular proteins, 9.4% were membrane proteins, but 77% were cytoplasmic proteins. Now, using ingenuity pathway analysis, we looked at potential dysregulated disease pathways in CF ductal cells and the upstream signaling pathways. Now, disease pathways included endocrine disorders and inflammatory response as being potential dysregulated pathways, and upstream canonical pathways leading to changes in the proteome included epithelial junction remodeling as to be positively impacted, and suppressed EIF2 signaling and oxidative phosphorylation that led to the differential proteome that we saw. Now, what we also did was look at top upstream regulators or regulatory pathways that could potentially cause this differential proteome we see in CF, and saw that TGF beta and Wnt signaling, shown here TGF beta and CTNNB1, which is beta catenin or Wnt effector, were a part of the upstream regulators. And it also included inflammatory pathways like mTOR. So shown are the contributing proteins that were a part of, that are targets of these respective pathways and are differentially secreted or are differentially expressed in the CF cells. Now, to conclude, loss of CFTR changes the CF ductal epithelial secretome and proteome. Now, second is alteration in the CF ductal epithelial secretion are potentially due to changes in TGF beta, Wnt, and BMP signaling. One key candidate that we saw was differentially secreted in CF ductal epithelia is IGF-BP7, which we also found to alter islet function. Loss of ductal cell CFTR can impact islet function via paracrine signals, and TGF beta signaling can be implicated in the change in CF ductal phenotype observed in CF pancreas. So with this, I'd like to thank all of my lab members where I did my PhD. I'd like to thank the funding sources and collaborators, the departments of anatomy and cell biology and biomedical engineering at University of Iowa, and the organizers of the webinar for giving me a chance. With this, I'd like to take any questions.